everyone, my name is Arthur and I'm a second year master's student in cell and molecular biology in Dr. Mark Chan's lab at San Francisco State University. And my research focuses on examining the effect metal toxicity has on vacuole inheritance and structure in Saccharomyces cerevisiae. So as many know, metals are necessary for biological function to occur, but when there's excess environmental metals present, they begin to non-specifically bind throughout the cell, which leads to disruption of these functions. And a novel discovery our lab made is that if you treat cells to lithium, we can actually inhibit the inheritance of the vacuole from the mother cell to the daughter bud. And the vacuole is very important because it carries out numerous functions like maintaining cellular homeostasis, carrying out derogation and detoxification, potentially storing metals, and the vacuole shares homology with the mammalian lysosome. Our lab has a model in how this inhibition occurs, and it is through the GSK3, YCK3, VPS41 pathway. So when lithium is absent, GSK3 phosphorylates YCK3, YCK3 then phosphorylates VPS41. When VPS41 is phosphorylated, vacuole fusion is inhibited, and we allow inheritance to occur. We need to inhibit this fusion event because if we don't, the vacuole and the mother cell will fragment, but then it'll refuse back together into one big vacuole, preventing the daughter bud from ever receiving anything. And what lithium does, it inhibits GSK3 and prevents this downstream phosphorylation event from occurring. And this is data collected by a previous lab member that made all of these discoveries. When we treat the wild-type cells to lithium, we get around 20 to 30% defective vacuole inheritance. In the control condition, it is only around 5%. If we delete VPS41, we see a recovery in inheritance, but if instead delete YCK3, we see a drastic increase in this defective inheritance. Also, if we take the VPS41 deletion strain and transform it with a VPS41 phosphomimic plasmid, so this is just a plasmid that is mimicking VPS41 always being phosphorylated, meaning vacuole fusion is always inhibited, we see a further recovery in inheritance to a level similar to that of the wild-type control condition. And if we were to assume our pathway is linear and the most downstream effector is VPS41, we are left with the question, can slowly phosphorylating VPS41 restore inheritance when YCK3 is absent? I also investigate how do lithium-magnesium mixtures impact inheritance. So it's been suggested that magnesium activates GSK3, but data collected by our lab suggests that magnesium inhibits inheritance, but to a lesser degree than what we see in lithium-treated cells. And we're left with the question, is the impact on inheritance additive if cells are treated to lithium and magnesium at once? I also investigate metal accumulation in the vacuole. Like I've mentioned, the vacuole may potentially store metals, but all that research has been done indirectly in the literature, and data collected by our lab suggests that vacuole size remains relatively the same regardless of excess environmental metals being present. So this questions the validity of the vacuole serving as a primary metal storage site. And I want to thank Dr. Mark Chan, the Chan Lab, the ARCS Foundation, the SEO Office, the NSF SCC CCC for making this research possible. Thank you.